we're going to use some ATP. Is this what you were talking about?
Yeah, is that
Okay,
like
it's just, I need to use the energy of 2 ATP to change this glucose to this.
but is it 2 ATP molecules? All right,
Yeah.
makes sense.
All right, so now I got my G3P back. So then I have to get the pyruvate to complete the glycolysis cycle. So in order to get to this molecule from this one, I've got to do some transactions. I'm going to make some NADH, right? And I'm going to make some ATP. Now I make 4 ATP, but I've used 2 ATP earlier. So in glycolysis, the whole thing, right? I only get 2 ATP out of it.
Okay.
All right, so now I've got my glycolysis done, I've got my pyruvate, now I've got some choices. I can breathe and do aerobic respiration, or I can hold my breath and hope that my cells can do anaerobic respiration long enough for me to live.
Nope.
All right,
Yeah.
so let's look at this first process first. All right, now you see a line. I'm taking my pyruvate from the cytoplasm and moving it into the mitochondria.
Yes,
This
of course.
only happens when oxygen is present. There's an enzyme that's responsible for bringing the pyruvate over to the mitochondria, and it kind of unlocks the door and lets it in. But that enzyme only does it when it has like its co-pilot, right, which is oxygen. Right? If oxygen isn't there, that enzyme just sits around and goes, I'm not doing nothing. Right? The same thing in some cells. This NADH also, there's an enzyme, but not all cells. This NADH can be transported into the mitochondria if there's oxygen present. Okay? This is where you see on the diagram where it says there's a range of ATP that can be made. If I can use this in the mitochondria, I can get the most out of my glucose. Okay? I'm going to use that to make ATP over here. Yes.
How can the NAD plus become NADH if it's binding with an H plus?
What you talk about, Will?
Like right here. How does 2 NAD plus become 2 NADH?
So what happens is from this G3P,
Yeah.
I'm going to strip off a hydrogen, for each one, each NAD, I'm
Yeah.
going to strip off a hydrogen and an electron. So I'm going to convert it from NAD
Oh,
plus
okay.
to NADH. So I'm doing that twice,
Like
one for each one. So I'm getting one off of here, I'm getting an NADH from this G3P, and I'm getting an NADH from this G3P. Okay?
Yeah.
All right. So if oxygen is present, so that's what I'm going to do here. Let's start zooming in here a little bit. I'm going to write here O2 present.
present.
Right? That's going to be aerobic respiration. AEROBIC. So I'm going to do aerobic respiration. Now, because I've done this before, and through trial and error over the years, what we discovered was it was a lot easier to just show one pyruvate going through the process. If we do you need a seat, don't you?
What?
If we did more than that, it got messy in here. So what I want to write is a little note up here. For each pyruvate. So this is for each pyruvate, which means I have to do it twice for one glucose molecule. Right? Okay. So oxygen is present. I'm going to bring my pyruvate molecule into the matrix of the mitochondria, the interior space here. Right? When it comes in, it's going to meet up with coenzyme A. And I'm just going to write CoA. So capital C, little o, capital A. I'm not going to write out coenzyme A too much, right? Now, when I meet up with my coenzyme A molecule, right, I'm going to break off a carbon dioxide, and I'm also going to make one NADH. So I'm going to break off a hydrogen electron. So I'm going to do an NAD plus becomes NADH here. Okay. Now, this carbon dioxide that I broke off, like all the carbon dioxides in this process, it's going to diffuse out of the mitochondria and into the cytoplasm of the cell, and then out of the cytoplasm of the cell through the cell membrane into the bloodstream, where then it's going to get to the lungs, and then you're going to exhale it, right? Get rid of it. So I'm going to write a little note up here so I don't forget this, right? CO2 diffuses out of the cell as a gas. Right? So CO2 is diffusing out of the cell as a gas. Right? Okay, so now I've manipulated my pyruvate to where I've removed a carbon, 2 oxygens, a hydrogen, and an electron, and that's given me this acetyl group that's left over that is attached to my CoA. Right? So this is acetyl, ACETYL, CoA. All right? I'm not going to write out coenzyme A, but that's what it is. Acetyl coenzyme A. Okay? All right. So now I've got my acetyl CoA, and it's going to come over to this molecule down here, this 4-carbon starter molecule. It's called oxaloacetate. OXALOACETATE. Right? Oxaloacetate. Now my oxaloacetate meets up with the acetyl-CoA. The CoA says later. Right? It drops off. It bonds the 2 carbons of the acetyl group to the oxaloacetate. The CoA is going to go back over here and interact with another pyruvate. So it's just an enzyme that's just doing this over and over again, back and forth, back and forth, back and forth. All right? Yes.
Then what exactly is the point of it?
To change it into an acetyl group from pyruvate and to bring it to the oxaloacetate and then make citric
Acid.
acid.
Oh, actually,
Hence
guess.
the name citric acid cycle. So Krebs cycle is also known as citric acid cycle. Right? So we're making the citric acid. So we took our 4 carbons and our 2 carbons and we made a 6-carbon citric acid.
Actually, guess.
Now,
Wow.
what we need to do in this Krebs cycle process is we've got to take our citric acid, all right, and we've got to get it back to oxaloacetate so we can do it again. Right? And so what we're going to do is we're going to harvest materials from the citric acid and use some of them later. Right? So what we're going to first do to our citric acid is take off a carbon dioxide, which is going to diffuse out of the cell just like the other one. Then we're going to make some NADH. So I'm going to take an NAD plus, and I'm going to make 1 NADH. So I've removed a hydrogen and carbon, 2 oxygens, and an electron from the citric acid. I have this new 5-carbon molecule, and here I'm going to remove a carbon dioxide again. Right? Now this carbon dioxide, this one, and the one in the transition phase that we talked about before, all 3 of those are going to diffuse out of the mitochondria. Now these 3 represent almost all of the carbon dioxide that you exhale all the time. Right? There's a fraction of a percentage of the carbon dioxide that you do exhale, though, that isn't from here. It's from your liver breaking things down. Right? So your liver does produce some carbon dioxide that goes into your bloodstream and you exhale. But most of it is coming out of the matrix of the mitochondria here. All right. So now after I take off that carbon dioxide, I'm going to make ATP. I'm going to take 1 ADP plus a phosphate group, and I'm going to make ATP. So 1 measly ATP per pyruvate. Right? I'm also going to make some more NADH. So I'm gonna take an NAD plus and turn it into an NADH. So I've removed another hydrogen and I've removed another electron. Now, this process is gonna get me to this 4-carbon molecule, right? Now, even though oxaloacetate is 4 carbons, this molecule still has too many hydrogens. So I've got to get them off to get back to my oxaloacetate. So I'm going to interact here with a new molecule, FAD. And FAD is like a big taxi cab. It's going to carry 2 hydrogens. So it becomes FADH2. Okay, so FADH2. But I still have a little bit more to go. All right, I've got to remove 1 more hydrogen and 1 more electron to get back to oxaloacetate. So I'm going to do that there. NAD plus is going to become NAD. So this is, and now I'm back to oxaloacetate, where I can meet up with the acetyl group again and become citric acid and go through the cycle again. So this is my Krebs cycle. Alright, more citric acid cycle. Alright, now my Krebs cycle doesn't get me much ATP. Right? For each glucose I'm only making 2, because this is per pyruvate. Right? I only get 2 measly ATP for every glucose I've converted in glycolysis. But I've made 1, 2, 3, 4, 5, 6 hydrogens are stored here as potential energy sources in my NADH and FADH2 that I've used. All right, that's 6 per pyruvate, which translates to 12 total for my glucose. How many hydrogens are in my glucose?
8 and 12.
12. So I'm going to use all of those hydrogens in that glucose to do work. Right? So we're going to do that work over here in the electron transport chain.
My man.
We're going to do some work. We're going to do chemiosmosis. So the first thing I'm going to do is I'm going to start off here, right at the beginning, with some NADH. So I'm going to take an NADH, and I'm going to strip it of its hydrogen and electron. So it's going to be back to NAD+. Now the NAD+ is going to be able to go back over here to the Krebs cycle and pick up another hydrogen and come back. So it's recycled. Right? That NAD+ in the mitochondria just keeps carrying hydrogens out of the Krebs cycle and bringing them over here. Right? So it's dropping off the hydrogen. The hydrogen loses its electron too, so I get a proton into the matrix here. And since I lost 2 electrons, they end up in this protein. Right? 2 electrons. Right? These 2 electrons are going to do some work. This is all not only stripping the NADH, but this protein also is a proton pump. So we're going to move protons from inside the matrix to the intermembrane space. Now, because I have 2 electrons, I'm going to be able to move 2 protons. As each electron zooms through, it's going to pull a proton in. So I get 2 protons on this side. Right, now, these electrons, yes?
Can you write the 2 protons as just 2H+?
You could if you want, but I want to make it look crowded. Right. So, these 2 electrons are going to move down the electron transport chain, or ETC. Right? Now, this is just the chain of proteins that we're looking at here. Now, the electrons get to this protein here, and they're going to interact with FADH2. So FADH2 is going to be stripped of its hydrogen, so it becomes FAD again. And that FAD is going to go back into the Krebs cycle, pick up 2 more hydrogen, and then come back. So it's just a little, like a larger taxi cab. It can carry more hydrogen than the NAD+. Okay? So, now, it's lost its hydrogen, but something's going to happen here. We're going to take off the electrons of that hydrogen too. So I really just have 2H+, right?
Wouldn't it be FADH plus then?
No, because it's 2 hydrogens, FADH2. Okay? Alright, so now, since I have 2 electrons coming from here, and 2 electrons coming into here, how many electrons are in this protein now? 4. So 4 electrons are now going to travel down my electron transport chain to this next protein. So I've got 4 electrons coming into this proton pump. Alright, so I'm going to take protons from inside the matrix and I'm going to move them across. Since I have 4 electrons, how many protons do I get to move?
4.
4. Alright, so I'm going to draw them in. H+, See, it's starting to get crowded on that side. Okay? Now, these 4 electrons continue to move down the chain, and they're going to end up here at this last protein. This one is also a proton pump. Okay, so I'm going to take protons from inside, and I'm going to transport them across into the intermembrane space. I get how many protons? 4. 4. Right, so I'm going to draw it in. H+. All right, so I'm creating a concentration gradient, a proton gradient. Right, I got more protons on this side than I do over here. So what do they want to do?
Move back.
They want to get back. They want to become equal. So their only escape route is going to be this guy over here. ATP synthase, exactly. So I've got a proton here that wants to get out. He's waiting at ATP synthase. SYNTHASE. Alright. At ATP synthase, my proton is going to move through the ATP synthase and come out into the matrix. Now, as the protons move through my ATP synthase, they're going to cause it to rotate, and that rotation is going to allow it to pick up an ADP plus a phosphate group, and we're going to make ATP. Now, you get about 1 per proton coming through, but it doesn't really work out that way, right? The general hypothesis on that is that as it spins, it may pick up an ADP and a phosphate. Sometimes it doesn't, so it's got to spin around again to do it. Okay, so it's not a 1-to-1 ratio. Yes?
What?
Over here? Right here. This
You
one?
mean on the
Am I missing something? Over here?
Yeah, with
Oh, you noticed something's wrong. Yes, I missed something. Yes, I know, I did that on purpose.
I was thinking you
Alright,
get to it.
so I dealt with my proton gradient here, but we have a problem. Because we have electrons coming down here and they're not going anywhere, right? This would be bad for the cell. If we built up electrons here, we'd get a charge and the cell would essentially electrocute itself. And it wouldn't be very successful, right? So what happens here is we have to have a final electron acceptor.
NADP reductase.
And what's the final electron acceptor in mitochondria? Do you guys remember?
NADP reductase.
It's not reductase.
Breathing.
Oxygen.
Oxygen.
Cyanide.
Oh yeah, that dude.
Cyanide.
So O2. So I'm going to take
We
O2.
breathe cyanide.
I'm going to take O2
You just
and
said
I'm going to add to it plus 4H plus. So all of the oxygen that you inhale, this is why you breathe. It goes here, alright? You breathe to rid yourself of that carbon dioxide from the transition phase and Krebs cycle, and you breathe to supply the mitochondria with oxygen at this location. Alright? So, now that oxygen, those O2, and those 4H plus, and those 4 electrons, are going to combine to make what?
Water.
Water. How much water?
Four.
Count up the-
Four.
what you got
Four.
there.
Four. One.
Two
Oh.
hydrogens
Two.
for every.
Three.
How many
Four.
oxygens
Five.
in water?
One.
One.
In one water molecule, how many oxygens are there?
One.
One.
There's two
So
oxygens.
how many waters could I possibly make in this transaction? Two. Each of these 4
Four.
electrons are going to fill each of these 4 hydrogens. They're going to all come together and we're going to get 2H2O. Alright? So I've got 2
C.
waters. Alright? So this is what happens to all that oxygen you breathe. It becomes water. Alright?
Good to know.
Okay?
I'm breathing to make water.
That's right, you are. You're breathing to make ATP.
So why do we need
Because
water?
if you don't have oxygen,
Because
the
if
electrons
we sweat it
stop
out
moving.
and then get
Alright? And when they stop moving, you
If I
stop making
breathe
the
enough, I
proton
won't need
gradient,
water.
and then you stop making ATP. Right? So that's why you need oxygen.
If I
But
breathe
now,
enough,
wait a
I won't
minute.
need water. What?
We've talked about ways that we can make ATP without oxygen. And it's
Fermentation.
called anaerobic respiration, and it requires a process called
Fermentation.
fermentation to keep it going. So let's look at that over here. Alright? We're going to look at it here. So if oxygen is absent, so I'm going to write O2 absent,
He didn't come to school today.
you won't come to school. That's right, and Melsick will probably cry. If oxygen is absent, alright, that's going to be anaerobic respiration. A-N-A-E-R-O-B-I-C. Anaerobic respiration. Now, in order to keep this going, you have to do a process that's called fermentation. So I'm going to write fermentation here. Alright. Now, came up with this handy dandy little drawing down here. Now what I did was I took that glycolysis box up at the top there and I shrunk it down even more. I got way down to the nitty gritty. What really is happening here? Alright? And so it turns out we're doing glycolysis and we're doing fermentation down here. So let's talk about, what's our first molecule in glycolysis that we need to deal with?
Glucose.
Glucose. So that's what this 6-carbon molecule is. It's C6H12O6, otherwise known as glucose. Alright? Now, we're going to take our glucose, we're going to break it down and do some stuff to it, and in the end, we're going to get out a whopping 2 ATP by taking 2 ADP and plus 2 phosphate groups, 2 extra phosphate groups. Oh my god, that 2 is terrible. 2 phosphate groups, and we are going to make 2 ATP. But in order to do that, we have to use some NADH. Alright? So we're going to take NAD plus, alright? And this is supposed to be an arrow, I realize. We're going to take 2 NAD pluses. Alright? And they are going to meet up here with this molecule, and we're going to create 2 NADHs. Alright? And we've got 2 pyruvate that we're making, right? Now, it turns out that in glycolysis, all this stuff that's going on up here, right? We've got all these things happening, right? And we're getting our ATP out of it, we're getting this. Right? It turns out that cells only have so many of these NAD plus molecules. right? They don't have an infinite supply of them. So if you turn off the oxygen, all right, you start to run out of NAD+. So you have to, in order to keep this going, to get these ATP being made on this side, you've got to have more NAD+. Once they're all filled and they're all NADH, this process stops. It grinds to a halt, and you are no longer doing glycolysis. You are no longer making these ATP. Yeah, and you will die. But some cells have an enzyme that allows you to recycle this, right? It's going to take the electron, right? It's going to change the final electron acceptor, all right? So it's going to take that hydrogen back off and the electron and make it back into NAD+. Okay? Now, we only have it for us in skeletal muscle. We don't have this in our brain cells, for example. So when your brain stops getting oxygen, this runs down, right? It comes to a stop, and then your brain's going to use this until it runs out of NAD+, and it's all filled up, and then it's going to just sit and wait. The cell in your brain is going to sit and wait, but it can only wait so long, and then it dies.
Big sad.
Big sad, yeah. So too many of them die,
You die.
you die too.
Respawn.
And then you got finna hit the
Bad.
respawn.
Wait, but what
Way
if you
too
put
many.
your bed down?
What?
No, no, no.
What are you doing in your lab? Why?
Kugar or volleyball?
We're not done.
Oh, what are you doing?
Alright, so, but our skeletal muscle has the ability to do fermentation. So, in human skeletal muscle and in mammalian skeletal muscle, what we do, in order to recycle this, you can see how we're recycling it here. That's all we're doing in fermentation. We're keep moving this NAD+ back so we can keep making these ATP. Right? So the top line here is what we do in our skeletal muscle. We have an enzyme that's going to interact with the pyruvate and the NADH, and it's going to take off the H and an electron and put it onto the pyruvate, and that molecule becomes 2 lactic acids. So that's how you make lactic acid. You just add 1 hydrogen and an electron to your pyruvate. That's lactic acid. All right. Now, other organisms, like yeast, for example, they don't have the same enzyme we do. They have 2 different enzymes. And those enzymes are going to allow it to do alcoholic fermentation.
Just get drunk,
So in
guys.
this case, so what happens is first an enzyme interacts with the pyruvate and removes a carbon dioxide. So since we have 2 pyruvate here, we're going to remove 2 CO2s. All right? And then the next enzyme interacts with it and puts on the hydrogen and electron, and that is to ethanol.
Ethanol.
Ethanol is big.
Ethanol molecules, all right? That's how you get your alcohol.
Yes.
So all we're doing is recycling my NAD+ to keep making these ATP. Now the process of glycolysis is actually relatively fast, so you can do it as long as you can do one of these processes and you have glucose. Oops, sorry. And you have glucose, you can keep doing this over and over and over and over and over again. Right? But you're going to be making lactic acid and ethanol. Yes?
How do you spell ethanol? Is it with A-N-D-O-L?
ETHANOL. That's a no. Yeah.
That looks like a D.
Yeah, it does look like a D. Alright, so. Have you ever run a mile?
No.
You're doing this. You're doing lactic acid fermentation. Alright,
I don't run.
so now in order to show you something that's important here, I'm going to do a little thing.
What's your margin?
Alright, I'm gonna do that, and then I'm gonna do this here. Alright, so over here on this side, this is glycolysis. This side is fermentation. Oops, what happened to the?
I wonder why did scientists
All right.
make the word so long?
Alright, so now when we look at this, in the glycolysis side over here, we're doing glycolysis, right? Fermentation is just the process of recycling that NAD plus molecule. So how the cell is able to do that. We showed two processes, lactic acid fermentation and alcoholic fermentation. Now, where is ATP produced? Is it produced during glycolysis or fermentation?
Glycolysis.
Wait, glycolysis.
Alright, so here, do this. Is ATP being produced here?
Yeah.
9,
Do you see ATP?
9.
What about here?
Yes.
Yay!
Wow.
So ATP is only produced during glycolysis.
Yosh.
No ATP is produced during fermentation. The role of fermentation is to recycle the NAD molecule. That's it. Yes, sir.
The ATP is used during fermentation, right?
No, it is not. This is an enzymatic reaction that requires no ATP.
Wow.
Just, wow.
The ATP is going to get used by the cell to do work.
Yeah.
So when you're running that mile, you're not getting enough oxygen into your mitochondria. The mitochondria is still doing work.
So what if we
but
are getting enough?
You're going to
oxygen?
burn, you're going to do glycolysis much faster. So you're going to use these ATP immediately.
So does
So
that
when
mean
you run and you start, you get to a certain point where you're breathing as hard as you can, and you just keep going in that mile. You keep going. You're actually doing this because you can get these ATP faster than you can get the ones from the mitochondria, and so you're producing lactic acid, which is why later on you're sore.
so we can't get drunk when we're running?
So you can't what?
We can't get drunk when we're running.
That's yeast,
No, we
mate.
can't get drunk when we run. Otherwise, I think as a species we'd be running constant.
I want to do
I think it would be the #1 sport.
How far could you ride this
In
dog?
classrooms, we just have treadmills.
Everyone would just like be high after.
Frat parties would be hop on the treadmill.
Probably not be a good thing for our species.
How would you even punish them?
Yes.
I forgot my question.
All right. Any other questions?
I just remembered.
What's